Louisiana Bioinformatics Training, a summer training program in bioinformatics in collaboration with the Louisiana Biomedical Research Network and the LSU Center for Computation and Technology. The course is being organized by Biotech and is going to be delivered in an innovative approach that was designed to allow hybrid access both online and offline. Project-based education in computational analytics for biomedical data and bioinformatics. Omics logic. The purpose of this course is to give a logic behind creating analysis pipelines and applying different methods to next-generation sequencing data, high throughput data that represents biological phenomenon that is being studied. And this course is being done with the help of the Tower Bioinformatics Research Center, which is a research center focused on the development, the design, and the development of the T-BIO.INFO platform at University of Haifa in Israel, and Biotech, a company based here in New Orleans in Louisiana. So we're pursuing three objectives in this course. Project-based learning, we're going to minimize the amount of theoretical knowledge that we're going to cover and focus on practical aspects on how to apply various analyses to different types of projects, data sets. We're going to be using research-grade tools that you will utilize, you're able to continue using them afterwards in real research projects. And the data sets that you are using now are real research data sets. And finally, we are looking for an environment where joint problem solving can help us see the problem from different perspectives and interact so that we can jointly solve problems that arise. In this course, we'll be utilizing the T-BIO.INFO platform, a platform that was designed to simplify the complexity of various types of public domain, open source, and proprietary methods, and provide, so to say, an open box, something that you can look into and use, as well as learn about how the platform works. So we're not necessarily focused on coding and knowing how to code and setting up the environment. We are focused on demonstrating how the tools work and then looking at projects that we can analyze and gain experience from in the application of these tools. We are combining theoretical knowledge with analysis logic. So we give an introduction. This course is going to be focused on oncology. And so the topics, the different projects that we're going to be utilizing are all oncology related. But of course they have a difference in the number of samples, the background of the samples, the data itself might be a little bit different here and there. So we're going to take these different variations of data sets, and then we're going to start by looking at suggested pipelines. Pipelines that assemble together a number of algorithms that are done in a sequential way, processing the raw data, converting it into a visual that we can interact with, and finally preparing and applying machine learning algorithms on these data sets. Throughout the preparation process, you will be seeing pop-ups, educational pop-ups with text, explanations, and links, and those will be included in the knowledge assessment quizzes, and finally an evaluation on how you're able to apply these different methods to different types of data. The course overview. First of all, we started with this kickoff workshop, and we are discussing here how to practically approach the different resources that we are providing, including courses, the platform, and the projects. And then we are going to spend a couple of weeks on Introduction to Bioinformatics, an online course that I will show in a second, and Transcriptomics 1. Another online course. Introduction to Bioinformatics covers big data challenges in biology and biomedicine, hypothesis-free analysis, and biomedical applications of bioinformatics. And then Transcriptomics 1 will deal with next-generation sequencing, how to take raw next-generation sequencing reads and align them as well as quantify the accumulated reads as a gene expression level and explore that data using PCA, principal component analysis, so that we can visualize that data. After these basic courses are completed, we are going to come here to the Center for Computation and Technology and have a 2-day workshop. Now, this 2-day workshop is going to give you some practical skills. We will go through a number of pipelines and compare results and look at some of the methods that we used in a pipeline. And the second part is going to be dedicated to an overview of projects, what types of projects we're going to be dealing with, and thinking about different approaches that could be relevant and interesting to individuals and groups of participants within this program. After that workshop, which is going to happen on June 25 and 26, we're going to spend a little more time going through Transcriptomics 2 and Transcriptomics 3. Both of these courses deal with quantification in a comparison. So the first option is differential gene expression, where you have a couple of groups and you compare the difference between gene expression within samples in those groups. But of course, there are limitations to that type of an approach. For example, sometimes you have a lot of variance within the group, or you might have more than 2 groups. And there's a challenge that could be overcome with methods that now could be described as machine learning methods or data mining methods, starting from unsupervised methods like PCA and clustering, and all the way to building a supervised model for classification like linear discriminatory analysis, support vector machines, and decision trees. After that, we will have another hands-on workshop, and by that time we are hoping that everybody has some level of project idea that they can communicate and discuss in a group setting, and we will make sure that everybody's on the right track. Spend some time reviewing some of the additional methods that we didn't have time to cover in the 2-day workshop, and start the independent project. And the independent project, we will cover that a little bit today, is an optional thing. You can either go with the project that was discussed in Transcriptomics 1, 2, and 3, which is the PDX project, or you can choose another one. We curated 2 of them and prepared them as educational exercises and in a structured way, or you can take a completely independent project, a data set that you bring. That might take more time, so we should plan accordingly. Finally, we'll do a project review where we will get together and do presentations of projects, as well as look at how the teams operated, evaluation, and provide certificates to all participants of the program. Now, the first big part of this program is using the online courses that you can find on EDU.T-BIO.INFO. These courses are structured, but they have text, keywords, images, and videos. And what you will find is that as you go through, there will be a lot of references to different methods, and those will be explained in charts, such as you see right here. And also, if you have any questions along the way, there's a chat box and you can always ask questions. Now, the second big part is going to be the platform itself, where you do analysis, and to do that analysis you will be provided with these projects. So these are data sets that come from the public domain. They have associated publications with them, and in the publications, authors describe what they use to analyze the data and how did they interpret their findings. Now, we will provide examples of how to do some of those methods or some alternative methods that are similar in the way that they work or perform a similar function. What are these projects? The first one is the whole transcriptome profiling of cancer tumors in mouse PDX models. An interesting project where we learn about the role of the microenvironment, the tumor microenvironment, and learn how to differentiate between the mouse and the human genes, and possibly correlate how various mouse and human genes and their expression levels are correlated with different subtypes of cancers. The second project is modeling precision treatment of breast cancer using multiple types of cell lines and therapeutic agents. An interesting project that takes multiple patient-derived cell lines and they are treated with various small molecule drugs, and then the efficacy of those drugs is measured using a measurement called IG50. And then there's a question of whether we can find specific molecular identification characteristics that could help us envision how these drugs might work better or worse in a patient. The third project deals with the tumor-associated macrophages. These are immune cells that could be recruited by the tumor to actually, instead of suppressing the tumor, assisting in its growth. And so we're going to be looking at the change and differentiation of these TAMs over time and look at how to deal with time course data. The fourth project is not provided as a prepared project, but it will be available as a publication and a dataset. It's a pan-cancer dataset that involves analysis of 12 cancer types and how to use different molecular data to perform classification. We will hold these workshops, and these are just examples of workshops that are either online or in person. Again, it's important to separate the things that you learn independently online is at your pace whenever you have time, and you can spend however much time is needed to complete everything. During the workshops, we will try to review some of those materials and address specific practical considerations around when, how, and why to use different methods. Of course, at the end of all of the program, you will receive the certificate of completion, and this will be from the Louisiana Biomedical Research Network. Throughout the course, we will ask you to fill out several forms that serve as pre- and post-assessment. We do ask that you identify yourself. We plan to use this data in a publication later on. But we also want to provide feedback to each one of you individually, as well as to the group and the organizers of this course. And we would like to learn from this experience and improve our courses, as well as provide you feedback about the skills and concepts that you've learned throughout this course. At the very end, you will be able to see the projects that individuals came up with. We will try and separate all of this cohort into a number of groups, and each one will be able to propose a project and then collaborate on that project together with other members of that group. And this is an example of what we did at University of Loyola in New Orleans, Loyola University in New Orleans, and some of our online students and what they thought about this project-based approach. Now, the next workshop that we are planning to do, this workshop will happen on June 25 and 26. Again, here at the the Center for Computation and Technology, the CCT, at LSU in Baton Rouge. And there we will start with an overview of some of the critical review of the approaches, how to compare different approaches, when to use different sections like pre-processing and different mapping, isoform construction, and expression level quantification methods. We will talk about differential gene expression, how to compare fold change between, so how to quantify change and see whether change is statistically significant between two groups, and then how to use machine learning methods to classify different samples. And finally, at the end, when you start learning about the projects, and we will start working on preparing ideas for analysis of the data. And these are just some examples of students getting feedback from faculty on their project ideas and some of the methods that they used in their analysis. So this is something that we plan to organize at the end so that everybody has an opportunity to both present and provide feedback to their peers, and faculty, of course, are welcome to participate in that as well. That's it for now. This is a short overview of what we will cover in the summer program. In addition to this video, we just posted another video on the different sites, the EDU site and the server site, how to use both of those, how to utilize the interface, what is the difference between the two, when you go to one link and the other. And finally, if you have any questions, you can definitely reach out to us. So everybody who is involved in organizing this course is listed right here. Of course, it's the LBRN folks, as well as the LSU Center for Computation and Technology, and Pine Biotech. So reach out to any of us with any questions or suggestions, recommendations. If something is not working, please do let us know. And again, thank you for joining.